neurotransmitters. The nerves, they are interconnected and the impulse travels through these nerves. Neurotransmitters, they are the chemicals which allow the transmission of signals from one neuron to the next across synapses. There are gaps or gap junctions between these neurons which is known as synapses, and the chemicals which allow the transmission of signals from one neuron to the next neuron. These chemical substances are known as neurotransmitters. They are found at the axon endings of motor neurons where they stimulate the muscle fibers. Neurons secrete different types of neurotransmitters based on where these nerves or the neurons are found. The neurotransmitter substances also keeps on changing. Each brain chemical works in widely spread but fairly specific brain locations and may have a different effect according to receptors of postsynaptic membrane. There may be either exciting one where it is conducted, where the impulse is conducted to the next neuron, then that is an exciting one where the action potential is being transferred to the next neuron. Or the opposite effect may also occur where it is inhibitory, where the postsynaptic neuron, the impulse stops at the postsynaptic neuron. They are the chemicals that communicate information throughout our brain and body, and the brain uses these neurotransmitters tell your heart to beat, your lungs to breathe, and your stomach to digest. All the functions of the body are performed by these substances which are known as neurotransmitters. They also affect based on the neurotransmitters that is being stimulated. This affects the mood, sleep, concentration, weight, and can cause adverse symptoms when they are out of balance. Neurotransmitter levels, they can be depleted by many ways. Stress, poor diet, neurotoxins, drugs, alcohol and caffeine usage can cause these levels to be out of optimal range, or these cause an imbalance. The neurotransmitters, they are stored in small sac-like structures called vesicles at the end of each axon. Every axon you have small vesicles, and these vesicles store the neurotransmitter substances. When an impulse or nerve signal reaches the end of the axon, when there is action potential and it reaches the axon region, the vesicles release the neurotransmitter into this synaptic gap. That is the gap between the presynaptic neuron and the postsynaptic neuron. You have a gap which is known as synaptic gap. So once the impulse reaches the first or the presynaptic neuron, the ending of presynaptic neuron, there that information is passed to the next neuron or the postsynaptic neuron through this neurotransmitter. So this picture gives an idea how it is being, the neurotransmitter, how it is secreted. Once the action potential reaches near this axon endings, there's a change in calcium. There's calcium influx occurs, and this calcium influx leads to these vesicles attaching to the axon endings, and this releases the neurotransmitter. Once the neurotransmitters are released from the presynaptic neuron, it reaches the postsynaptic membrane, the postsynaptic membrane of the postsynaptic neuron, and binds to the receptors. And once it binds to the receptor is determined whether this impulse has to be transmitted by the next neuron or the postsynaptic neuron, or whether it has to be inhibited, whether it is an excitatory one or whether it is a inhibitory one. That is determined by the receptors present on the postsynaptic neuron. Enzymes concerned with the synthesis of neurotransmitters are present both in the cell body and in the nerve ending. A portion of neurotransmitter is produced in the cell body and transported to the nerve ending. Usually the neurotransmitters, they are synthesized within the cell body and then they are transported by these vesicles to the nerve endings. Neurotransmitters, they function by changing the permeability of the cell membrane to various ions such as sodium and potassium when an action potential takes place. If an excess of sodium ions flow into the nerve cell, an impulse is generated, and if an excess of potassium ions flow out, the impulse is inhibited. The acquired knowledge about the neurotransmitters has led to the development of successful products for many brain disorders including epilepsy, schizophrenia, Parkinson's disease, depression, anxiety disorders and migraine. Even these are all imbalance in these neurotransmitters. So there are certain barriers. The blood-brain barrier prevents the entry of these neurotransmitters freely, but many disorders have been cleared with the help when we are studying in depth about these neurotransmitters. It has led to successful products for these disorders. Types of neurotransmitters, I told you there are two types, inhibitory as well as excitatory. One, excitatory neurotransmitters, they are what stimulate the brain, or those that are carried by the neurons. Those that calm the brain and help create balance are called inhibitory one. Inhibitory neurotransmitters actually balance mood and are easily depleted when the excitatory neurotransmitters are overactive. Neurotransmitters can be broadly split into three groups. They are either amino acid 1's glutamate, GABA 1, the gamma aminobutyric acid, the neuropeptides, that is vasopressin, somatostatin and neurotensin, and a rich class of monoamines, epinephrine, norepinephrine, dopamine, serotonin, acetylcholine as well as melatonin. Acetylcholine, this was the first neurotransmitter that was discovered, and it is responsible for much of the stimulation of muscles. At the neuromuscular junction, it is acetylcholine that is the neurotransmitter, and it's also present in the muscles of the gastrointestinal system. It is also found in sensory neurons and in the autonomic nervous system, and as a part in scheduling rapid eye movement, dream or the sleep. So this is how acetylcholine works. Acetylcholine, acetyl coenzyme A combines with choline. Acetyl coenzyme A combines with choline, and this gets converted to acetylcholine, where it is release of coenzyme A. Coenzyme A gets released, and this reaction takes place in the presence of choline acetyl transferase. So you get two products there. When acetyl coenzyme A and choline react in the presence of choline acetyl transferase, you get two products. It's a coenzyme A as well as acetylcholine. Acetyl coenzyme A and choline join. Acetylcholine is formed. Acetylcholine is packaged up to leave the axon. The acetylcholine leaves the neuron. Acetylcholine reaches the outer side of the synapse and binds to the receptor, causing the message to be carried forward. And after the message is carried from the presynaptic neuron to the postsynaptic membrane, acetylcholinesterase is released into the synapse. The acetylcholinesterase, what it does is, it breaks down acetylcholine and inactivates it. Whatever excess acetylcholine is found in the synapse or the synaptic gap, that is actually inactivated by the presence of acetylcholinesterase. Whatever binds to receptor that does the activity, some portion of acetylcholine that is reabsorbed by the presynaptic neuron, as well as it gets split, gets inactivated with the help of acetylcholine esterase, which splits it. Acetylcholine actually it is synthesized in liver. It is transmitted within the cholinergic. The neurons that actually the neurotransmitter is the acetylcholine. Those neurons are known as cholinergic neurons. And these are transmitted within cholinergic pathways that are concentrated mainly in specific regions of the brainstem and are thought to be involved in cognitive function, especially memory. Severe damage to these pathways is the probable cause for Alzheimer's disease. There's a link between acetylcholine and Alzheimer's disease. There is something on the order of a 90% loss of acetylcholine in the brains of people suffering from Alzheimer's, which is a major cause of senility. Outside the brain, the activity is different. Acetylcholine is the main neurotransmitter in the parasympathetic nervous system, which controls heart rate, digestion, secretion of saliva, and bladder function. Lower alpha and gamma motor neurons, all autonomic nervous system preganglionic neurons, all parasympathetic postganglionic neurons, sympathetic postganglionic neurons innervating sweat glands. There, every such places you find acetylcholine as the neurotransmitter. And these are cholinergic receptors, activate mainly sodium channels found in neuromyel junctions found in autonomic ganglia and blocked by Q. Norepinephrine. Usually norepinephrine's are inhibitory one, and these are released by adrenergic neurons. These neurons, adrenaline, since they secrete the neurotransmitter adrenaline, they are known as adrenergic neurons, and 90% they do not form synapses. Accounts for slow acting and long lasting effects for its release. It is actually norepinephrine. It is considered as a modulator that sets brain tone, suppresses irrelevant stimuli since it's a inhibitory one, enhances relevant stimuli and modifies behavior, arousal, degree of alertness, ECG activity and sleep. It plays a major role in mood as well as memory and is a key player in hormone regulation as well as homeostasis. Epinephrine, usually the opposite of norepinephrine, usually excitatory, found in neurons of lower brainstem, tegmentum. Dopamine is also usually inhibitory. Catecholamine, neurotransmitter, neurohormone released by hypothalamus, the only neurohormone released by the hypothalamus. Inhibit release of prolactin from pituitary, and it's present in neurons of the tegmental area. Dopaminergic neurons, those neurons are known as dopaminergic neurons since they secrete the neurotransmitter dopamine. Loss of inhibitory influences may be associated with Parkinson's disease. Serotonin, it's also known as 5-hydroxytryptamine and only present in cell bodies of neurons located primarily in enterochromaffin cells, gastrointestinal tract, platelets, central nervous system of animals, and the serotonin is known as happiness hormone. Though it is not a hormone, it is a neurotransmitter, and the nerves that use this serotonin for conduction of impulse are known as serotonergic neurons. Electroencephalogram or EEG. electroencephalography is a technique that records the electrical activity of the brain, just like how ECG records the cardiac one. Here it is the brain waves that are being recorded. During an electroencephalography test, small electrodes like cup or disc type, they are placed on the scalp at different regions of our head, and they pick up the brain's electrical signals and they send them to the machine called electroencephalogram, where these signals are recorded. These signals are recorded as waves. These signals are recorded by the computer. On the monitor you could see the waves moving. And the EEG, it measures activity of large number of neurons, and this was first recorded by Hans Berger in 1929. They are, you don't feel, it's just kept on top of your scalp and there's no pain. It doesn't interfere with any of your normal activities. It just records the brain waves under your brain, that is amplified, and that is recorded on an electroencephalogram. The electrodes measure voltage differences at the scalp in the microvolt range. The wave frequency shows the number of oscillations of the waves from its maximum amplitude to its minimum amplitude and back to its maximum amplitude. The movement from maximum amplitude to minimum amplitude and back, that is recorded as waves, which is displayed on a monitor, and this frequency is recorded in terms of hertz. And the group of neurons in hypothalamus generate the rhythm, which is conducted through nerve fibers from thalamus to the cortex region. And EEG is mainly used when there is a need to diagnose. Mainly it is used for epilepsy, some injury has occurred, like encephalitis, dementia, head injuries, brain tumors, hemorrhages. During those times, the waves, there's a change in these waves, and EEG could help us to identify the damage that is caused to brain. So based on the wave or the patterns, you could easily identify the region which is not working. When recording brainwaves, the electrodes are attached to scalp, low resistance connection connected to banks of amplifiers, and recording device is there which has a few wires, and at the tip of the wires you have the electrodes, and these electrodes actually they are connected to our scalp, and then the waves they are amplified and they are recorded, and the electrode pairs are there. Usually they measure different brain regions. So this is how a scalp is kept. This is how the electrode is kept on the scalp. So this measures the different activity that lies beneath, how the impulse is being carried, how the neurons work. This is recorded in the form of certain waves. And these EEG rhythms, categorization on rhythms, they are divided into different waves. Beta ones greater than 14 hertz activated. Cortex alpha, you have the frequency between 8 to 13 hertz, quiet waking state. And theta 4 to 7 hertz, some sleep states. Delta, deep sleep. When you are in deep sleep, it's the delta wave that is being recorded. Alpha waves seen in all age groups but are most common in adults, most marked in the parieto-occipital area, occur rhythmically on both sides of the head but often slightly higher in the amplitude on the non-dominant side, especially in right-handed individuals. So this is the alpha wave, how it looks. They disappears normally with attention. If you are listening to something, mental, arithmetic, stress, If you are in some state of rest, then it's the alpha wave that comes in, and this becomes replaced with irregular low voltage activity desynchronization, as it represents breakup of synchronized neuronal activity, usually of high amplitude, that is between 10 to 100 microvolts. Beta waves seen in all age groups, small in amplitude, usually symmetric and more evident anteriorly. When people think and become attentive, when you are listening to something and all, it is the beta waves that actually work. And it's seen during rapid eye movement, sleep. The frequency is less than 13 hertz and low amplitude, that is somewhere around 10 microvolts. So this is the wave pattern of beta waves. Theta waves normally seen during second and third stage of sleep at any age. And in awake adults, these waves are abnormal if they occur in excess, occur during emotional stress. Theta and delta waves are known collectively as slow waves, and it has a high amplitude which is of 100 microvolt. So this is the theta wave, how it looks like. Delta waves, just like theta wave, it's a slow wave, has a frequency of less than 3 hertz or less, normally seen in deep sleep in adults as well as in infants and children, and abnormal in the awake adult, have the largest amplitude of all waves. And if local pathology occurs, it's the delta waves